Sarah, can you tell us about this new threat, bacterial leaf streak, and why it's on your radar in 2023?
Yes, so bacterial leaf streak has been an issue for the last 2 or 3 years across the prairies. And up until this fall, there was no test for it. And it was something that we were watching and monitoring. So, with the assistance of Alberta Ag and Agriculture Canada, the research station in Lacombe and Brooks, it was decided that we develop this test. It's an issue because it spreads very, very quickly under ideal conditions, and it has been reported to cause fairly significant yield losses, although we don't know a lot of information about that. But if you look on the internet, it's anywhere from 10 to 40%. The reason it causes yield losses is because it affects the photosynthesis on the leaves, because the disease presents itself as a sort of yellow streak that follows parallel to the veins in the leaf. And in many cases, you'll see a yellow exudate, which is kind of like this water splashed marking, which is known as the bacterial ooze. It spreads very, very quickly. It's been known to double in intensity over a 24-hour period, given if the conditions are right. And what we're seeing is that in irrigated areas, particularly down south here in Alberta, where there is a lot of crop grown under irrigation, it's the water, it's the humid conditions that can ramp the situation up. And obviously, turning the water off and, arresting it at that point can help significantly.
Now, you're involved in some research trials regarding this. Can you tell us a little bit about those trials?
Yes. So we're working with Dr. Kelly Turkington at the research station in Lacombe. And initially, we contacted him because we did a soft launch with this disease testing. And through that, we were talking to customers who were sending in samples. So to date, we've had about 200 samples, and most of them are coming back positive with infection levels anywhere from low to high risk. And we decided, because there's very little known about it and how we can manage it, that we should get into some experimental work and do some research and find out what the actual DNA test means when we find it, and actually find out what the threshold is on our tests that are going to cause, significant issues in terms of yield losses. So we were going to go to the field, but it's been decided now that we'll do some growth chamber work. So that will involve taking samples from here with our client's permission, with various levels of infection, and doing a seed wash. So taking a dilution and plating it out and seeing, if it's viable, particularly that it's viable. You cannot identify the pathogen on a plate. It's always through DNA testing. And we'll grow out the cereals to a 2 to 3 leaf stage and see what those levels of infection, so anywhere from low, moderate to high, can create. So obviously using not detected as well, just to see that in actual fact that is the case. And we're going to run with possibly 2 temperature regimes, planting at 5 degrees and 25 degrees. The theory being that if we plant earlier, perhaps because pathogens don't like being seeded into cold temperatures, we might be able to see a reduction. So we'll do some comparisons between the 5 degrees and the 25-degree planting.
Now, Sarah, you've developed a molecular test for bacterial leaf streak. How does that provide growers with a tool that they can really use and get useful results from?
At this point, we're not saying to anyone, do not use this seed. Use best management practices until we know what the threshold is with the level of disease. So in terms of best management practices, the first thing is get your seed tested, find out what level of infection there is. Is it viable? And that would be potentially through doing a plate test. Then, in terms of seeding, seed early. We know that from previous situations like fusarium. If you seed earlier, it will suppress the disease and allow the seedling to get established. Sanitize all equipment. Do not move from field to field unless you've incorporated some kind of sanitation. That would be getting rid of things like the seed that's left in, say, the combine and debris and that kind of thing. Crop rotation. Try and avoid going into a field that has been previously seeded to a cereal crop. It's particularly infectious in terms of affecting other cereal crops. So obviously wheat, barley, durum and rye, but also wild grasses and other grasses. So, stay away from that if possible. And scout. Keep scouting. Look particularly around when there's been a weather event like high winds, rain, that type of thing, because it's thought that if it's in the area, you will definitely see it after a weather event. And that's when, if you're under irrigation, particularly with durum, we've noted, turn that water off and start to manage the water as well.
Before I let you go, I just like to emphasize the power of molecular testing. How does molecular testing, like you guys are doing, really change the game when it comes to not just bacterial leaf streak, but other threats like wheat midge, soybean cyst nematode, etc.?
Well, first of all, DNA testing has been extremely useful. It's very sensitive and it's accurate and it's fast. And producers today are looking for a very timely turnaround. So with anything that we do in terms of incorporating DNA testing into the laboratory, it's been very, very useful. And actually with the midge, for example, it's the way that we monitor the refuge. So the refuge is hugely important in terms of managing that particular variety and making sure that we're protecting the only gene that we have for midge tolerance, which is the SM1. So the DNA test allows us to test quickly and monitor any movements within that variety, and if there's been some extensive feeding and that type of thing, and it's changed up the level of refuge that's needed. So that's one of the things we use the DNA for. The soybean cyst nematode, we haven't done any testing. We've got the method, we've got the protocol. We know that it's just a matter of time before we start seeing it in southern Manitoba, because it largely affects soybeans, and that's a huge production area. And again, with the DNA, we can look at any level of the life cycle of the nematode, from the egg to the cyst. So it's important that we're able to detect that it's actually the soybean cyst nematode that's actually present. And then the Parma amaranth, that's probably the first one that's to come into the actual seed testing realm. So Parma amaranth looks very much like our native red root pigweed. And so you can't tell the difference between the Parma amaranth and the red root pigweed. And there's a lot of, red root pigweed that we see in samples all across Canada. And Parma amaranth is, has been moving quite quickly north, and it is herbicide resistant. So it's important that we find it and we manage it. So with the DNA test, if there's any likelihood that we suspect that we've got Parma amaranth in a sample, it's sent to the lab for the DNA testing. And there's lots of countries that don't want it. So, we've got to be able to prove that it's not in that sample that we're testing at that time that's going to be exported.
Thank you so much, Sarah, for your time. Have a great day.
You too, Mark. Always good to see you.